Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Ever get the feeling that our bodies are full of secrets? Like things that seem to do one job, but then it turns out they've got this whole other side gig going on.
Yeah, totally.
Well, think about this. Imagine this tiny messenger in your body that usually spends its time controlling your appetite, right? Turns out it might also be deeply involved in how a really deadly pancreatic cancer actually spreads.
Huh.
Wild, right? So today we are diving deep into some recent research that uncovered this pretty unexpected connection.
That sounds fascinating.
It is, it really is. So we are focusing on what's called pancreatic cancer metastasis, which basically means how the cancer spreads from the pancreas to other parts of the body.
Right.
And at the heart of this whole thing is a molecule called neuropeptide Y and one of its receptors.
Neuropeptide Y receptor 1.
That's the one. And to guide us through this, we've got this really cool scientific paper called Targeting the Neuropeptide Y/Neuropeptide Y Receptor 1 Signaling Axis in Mutant p53-Dependent Pancreatic Cancer Impairs Metastasis. And to give us a bit more context on the whole metabolism angle, we've also got a health news excerpt that talks about metabolic factors in pancreatic cancer research. But don't worry, we're going to really focus on those detailed findings from the scientific study for you.
Sounds good.
Yeah, our goal is to break down the really important insights from this research in a way that makes sense so you can understand this potential new way of tackling this really challenging disease.
Absolutely, without getting bogged down in the jargon.
Exactly, yeah. So before we get too deep into the nitty-gritty, I think it's really important to understand why stopping pancreatic cancer from spreading is such a big deal.
Right, it's crucial.
Yeah. So as you probably know, pancreatic cancer, it's a really tough one. It's super aggressive.
Unfortunately, yeah.
And the really scary thing is over 80% of people are diagnosed after the cancer has already started spreading.
Wow.
Which often means that surgery, which could potentially be curative, it's just not an option anymore. And sadly, once the cancer has spread to other parts of the body, the chances of surviving for a long time, they plummet.
So finding ways to stop it from spreading in the first place, that's the key.
Exactly. That is absolutely the key. And this is where our star molecule, neuropeptide Y, enters the scene. So, what made researchers even think this little guy might be involved in all of this?
Well, neuropeptide Y, it's already known to be kind of a troublemaker. Like it's involved in the growth and spread of tumors in several other types of cancer.
Oh, really?
Yeah, but its role in pancreatic cancer specifically, that was still kind of a mystery. So the researchers, they were on this hunt for new clues, like what is driving this aggressive spread in pancreatic cancer?
Makes sense.
And neuropeptide Y, it just seemed like this really promising lead to follow.
So how did they actually go about investigating this in the lab? Like, what did their experiments look like?
Okay, so they started with these specially bred mice that are designed to develop pancreatic cancer in ways that, closely mimic how it happens in humans.
Right.
And they specifically focused on two different mouse models with distinct genetic makeups.
Okay.
The first model is called the KP-FLOX-C model. So these mice have a mutation in a gene called CROS.
Okay.
And they also have this loss of function in another super important gene that normally protects against cancer called P53.
P53, yeah.
Now, here's the interesting thing. This particular model, it doesn't tend to spread as much.
Huh, okay.
The cancer, it tends to stay put more often.
Interesting. So what about the second model? How's that different?
Okay, so the second model is called the KPR172HC model. And these mice, they also have that same CROS mutation.
Okay.
But instead of the P53 gene being totally switched off, they've got this specific mutated form of the P53 protein. It's called P53R172H.
Okay.
Now, it might sound like a tiny difference, like who cares, right?
Yeah.
But trust me, it makes a huge difference. Because this KPR172HC model, it's way more prone to the cancer going rogue and spreading, especially to the liver. So having these two contrasting models, it was like this perfect setup. They could compare them side by side.
See what's going on, yeah?
Exactly. And figure out what might be driving that spread in one, but not the other.
Gotcha. So they've got these two models, one that spreads like crazy, one that's a bit more chill. What did they actually see happening with neuropeptide Y in these mice?
Okay, so here's the big reveal. They found that neuropeptide Y and its receptor, which is called NPY1R in mice, which is very, very similar to our human neuropeptide Y receptor 1, they were significantly more active in the tumors of those highly metastatic KPR172HC mice
Oh, really?
compared to both normal pancreas tissue, so like the healthy tissue, and the tumors of those less metastatic KP-Floxy mice.
So it's like the cancer cells in the aggressive model, they were just pumping out way more of this neuropeptide Y signal.
Yeah, it's like they were going one with it, like, let's spread, let's go.
Wow.
And get this, they even found the neuropeptide Y protein itself in the liver metastases of those KPR172HC mice, which suggests it was right there at the scene of the crime, where the cancer was setting up shop.
Okay, so now for everyone listening, I bet you're thinking, okay, so there's this correlation, but is it just a coincidence? Is neuropeptide Y really a key player in all of this, or is it just kind of hanging out there? So what did the researchers do next to really figure that out?
Right. They had to make sure it wasn't just a bystander.
Exactly.
So to see if this increased activity of neuropeptide Y and its receptor was actually a driving force, they decided to look at human pancreatic cancer samples.
Okay.
They dug into data from this massive database called the Cancer Genome Atlas. You might have heard of it. It's not referred to as TCGA.
Yeah, TCGA.
So what they discovered was that the levels of those genetic instructions, those messenger RNA levels for neuropeptide Y, they were also through the roof in human pancreatic ductal adenocarcinoma tumors.
And just to remind everyone listening, that's the most common type of pancreatic cancer.
Exactly. And these levels were way higher in those tumors compared to normal pancreas tissue. And when they zoomed in on those 4 different receptors that neuropeptide Y can bind to, guess what? Neuropeptide Y receptor 1, it stole the show. It was the most active one in human pancreatic ductal adenocarcinoma.
So basically what they saw in the mice, they saw it again in human samples.
Yep. More aggressive cancer, higher activity of neuropeptide Y and its receptor.
That's a pretty strong hint that it's not just some innocent bystander.
It's starting to look like a prime suspect.
Right. So how did they take that next step and actually test whether neuropeptide Y receptor 1 is really involved in the actual spreading of the cancer?
All right, so this is where it gets even cooler. They went back to their highly metastatic KPR172HC mouse model.
Okay.
And they used some fancy genetic techniques to basically dial down or completely shut off the activity of the NPY1R receptor.
So that's the mouse version of our neuropeptide Y receptor 1.
Exactly. And they did this in 2 different ways. So first, they did this knockout thing specifically in the pancreas. So the receptor was only inactivated in the pancreatic cells. And then they did a whole body knockout where they inactivated the receptor throughout the entire mouse.
Oh, wow.
So this way, they could see if the effect was really localized to the pancreas or if it had, these broader implications.
That's really clever.
Yeah, it's a really cool way to pinpoint what's going on.
Absolutely. So what happened when they messed with this receptor in the mice? Like, did it actually make a difference?
All right, drum roll, please.
Drum roll.
In both the pancreas-specific and the whole-body knockout models, they saw a significant reduction in how much that cancer spread to the liver.
Whoa.
Which, as we know, that's a major hotspot for pancreatic cancer metastasis in humans.
Yeah, for sure.
But here's another interesting twist. They noticed that even though metastasis was way down, the mice's overall survival, it didn't really change that much.
Huh.
So it seems like the receptor's main job in this model is to really drive that spread of the cancer rather than the initial growth of the tumor itself.
That's a really important distinction. Because for those of you listening, what this suggests is that if you target this receptor, you might be able to really effectively stop that cancer from spreading.
Precisely.
That's amazing. So if blocking the receptor genetically had this effect, the next logical step would be to see if they could do the same thing with a drug, right?
You got it. So to see if they could mimic these results using a more, drug-based approach, they used a specific neuropeptide Y receptor 1 inhibitor. It's called BIBO33304.
BIBO3304.
Catchy name, right? So first they took this inhibitor for a test drive in the lab, with cell cultures.
Okay.
And they found that BIBO3304 really put the brakes on the movement of those highly metastatic KPR172HC cancer cells. Like, it significantly reduced their motility.
Wow.
And they grew these cells on this special 3-dimensional scaffold, like this little platform that's designed to mimic the environment that surrounds tumors.
Huh, interesting.
And here's another interesting thing. This inhibitor, it didn't really have the same effect on those less aggressive KP-Floxy cells.
So it seems like it was specifically targeting those more aggressive cells.
Yeah, like it had a preference for the troublemakers.
That's amazing. So They saw this effect in the lab, but what happened when they actually gave this drug to living mice?
All right, so for this, they used a slightly different mouse model of metastasis.
Okay.
They injected those highly metastatic KPR172HC cancer cells right into the spleen of the mice,
Yeah. Okay.
which, by the way, that's a route that usually leads to the cancer spreading to the liver.
Right.
And then they treated these mice with R-star inhibitor, 503-304. And guess what? They saw a major decrease in both the number and the size of the tumors that popped up in the liver.
Wow.
And this was true whether they started the treatment pretty early in the metastasis game, like at day 5 after they injected the cancer cells,
Okay.
or later at day 12 after some spreading had already happened.
So even if it's already started spreading, this drug could still be helpful.
That's what it seems like, yeah. Pretty remarkable, right?
Yeah, absolutely. So it sounds like this inhibitor is really messing with the whole metastasis process in these mice. Did they get any clues about how it's actually working? Like what's it doing at the molecular level?
Yeah, they did some digging, and it looks like early treatment with 503-304, it's at least partly reducing metastasis by basically slowing down the growth of those cancer cells once they reach the liver.
Oh, interesting.
So it's not just stopping them from moving, it's also making it harder for them to establish those new tumors in other organs.
So for those of you listening, this research, it's pretty exciting stuff. It points to a whole new way to think about and potentially treat this incredibly challenging cancer. So what are the big takeaways here? Like, what does this mean for future research and treatment?
Well, I think the most important takeaway is that this research shines a spotlight on the neuropeptide Y and neuropeptide Y receptor, one signaling pathway as this, like previously hidden target for stopping or slowing down the spread of pancreatic cancer.
Wow.
And this is particularly true in cases where the p53 gene is mutated.
That's a huge deal, discovering a whole new target. What could this mean for actual treatments for patients down the line?
Okay, so the fact that the neuropeptide Y receptor 1 inhibitor worked so well in these preclinical mouse models, really gets you thinking. It's like, hey, maybe we can develop similar drugs for humans, right?
Yeah, absolutely.
And another cool thing is that the studies show that even just dialing down the receptor's activity a bit, like not even totally blocking it, still had a noticeable effect in mice.
Huh.
Which suggests that we might not need to completely shut down the receptor in humans to see some benefits.
And that could mean fewer side effects.
Exactly, less collateral damage.
That's amazing. So how might a drug like this fit into the current treatment landscape for pancreatic cancer? Like, what's the big picture here?
Well, the researchers are thinking that inhibiting neuropeptide Y receptor 1 could be this really valuable addition to the treatments that are already out there.
Okay.
Like those standard chemotherapy regimens that often include a drug called gemcitabine.
Gemcitabine.
It's possible that using this new approach alongside those existing treatments could really boost their effectiveness, make them even better at preventing that cancer from spreading.
That would be amazing. Now, we talked a bit earlier about a health news excerpt that mentioned how metabolic factors play a role in pancreatic cancer.
Right.
So how does this research fit into that bigger picture, the whole metabolism connection?
Yeah, that's a really important point. So this study, it really focused on the direct impact of the neuropeptide Y and neuropeptide Y receptor, one pathway on the cancer cells themselves.
Right.
But we got to remember that neuropeptide Y is not just hanging out there doing nothing. Like it has this other job. It's involved in regulating things like our appetite and energy balance.
Oh, right.
Which are like these core metabolic processes in our bodies. So this research, it adds another piece to this giant puzzle of how metabolic factors can mess with the development and progression of pancreatic cancer. It's like, okay, now we have this specific link between a molecule that's involved in metabolism and the spread of the disease.
So it's like this whole hidden network that we're starting to unravel.
Exactly.
Now, you also mentioned this interesting observation about tissue wasting in the mouse models, and that's something that really impacts a lot of cancer patients, right?
Absolutely. So in the mouse models where they knocked out that NPY1R gene, they noticed that the mice actually gained weight. Like their fat and muscle tissue increased.
Wow, that's wild.
Yeah, and it's especially interesting because pancreatic cancer, it's notorious for causing severe weight loss and muscle wasting.
Yeah.
Doctors call it cachexia.
Cachexia.
And it can really affect a patient's quality of life, their ability to handle treatments, everything.
Absolutely.
So the fact that drugs targeting this pathway might also help fight this really debilitating tissue wasting, well, that's definitely worth exploring further.
Yeah, for sure. Now, this research, it's super promising, but like with all scientific studies, we got to be realistic, right? There are always limitations in areas where more research is needed.
Yeah.
So what are some of the caveats here?
Totally. So first off, it's important to remember that most of this research, it happened in mouse models and cell cultures in the lab,
Right.
which is great. Those preclinical studies are super important. They lay the groundwork.
Absolutely.
But we can't just assume that what we see in mice will translate perfectly to humans.
Of course not.
There could be differences in how diseases progress and how they respond to treatments.
So basically, we need more research to confirm these findings in humans.
Exactly. We need those clinical trials.
Now, what about that specific P53 mutation that was involved in those mouse models that showed such dramatic effects?
Right. So that's another important thing to keep in mind. The most powerful effects on metastasis, they were seen in those mouse models that had that specific R172H variant of the mutated P53 protein. But human pancreatic cancers, they can have all sorts of different P53 mutations or even no P53 function at all.
Oh, wow.
So it's crucial to figure out whether the neuropeptide Y and neuropeptide Y receptor 1 pathway,
Yeah.
if it behaves the same way in pancreatic cancers with these other genetic profiles.
So basically, more research is needed to see if this approach will work for all types of pancreatic cancer.
Exactly.
Now, one last question. Are the researchers totally sure about exactly how this pathway is actually driving the spread of the cancer? Like, do they have all the details figured out?
So they've got some really good leads, but they're still piecing it all together. The studies show that inhibiting neuropeptide Y receptor 1, it definitely reduces the movement and growth of those cancer cells.
Okay.
But the exact step-by-step process, the precise molecular mechanisms by which this neuropeptide Y system is promoting metastasis, that still needs more investigation. Like, they found a bunch of other genes and proteins that were affected when they knocked out that NPY receptor in the tumors, which suggests that there's this whole complex network of downstream effects going on.
Oh, wow.
So, yeah, there's more work to be done to fully understand all the intricate details.
So what's the bottom line? Like, what did the researchers themselves have to say about the significance of their work?
Well, they were definitely excited. They highlighted the fact that identifying neuropeptide Y receptor 1 as this potential drug target, it's a big deal. Like, this is a whole new avenue for pancreatic cancer metastasis.
Yeah.
And they believe that targeting this pathway could be this super effective new strategy to stop the spread of this disease.
That's amazing.
And they're really pushing for more research, those clinical trials, to really assess this approach alongside the existing treatments.
Now, they also mentioned that this specific inhibitor they used, it doesn't really cross into the brain that easily.
Right.
Why is that important? Like, what's the significance of that?
So that could actually be a big plus. That inhibitor they used, BIBO3304, it's not very good at getting past the blood-brain barrier.
Okay.
And that barrier is like this protective shield for the brain. It keeps a lot of substances in the bloodstream from just waltzing in.
Yeah.
So because of this, the drug is less likely to mess with how the receptor works in the central nervous system.
Okay.
Like its role in controlling things like appetite and energy use.
Makes sense.
And those can be really troublesome side effects for cancer patients who are already dealing with things like weight loss, fatigue, changes in appetite. So having a treatment that doesn't make those issues worse, well, that's a huge win.
Absolutely. So for you listening, this deep dive, it's really revealed a potential new direction in the fight against pancreatic cancer. And it makes you think, if a system that's involved in something as basic as appetite control can also be pulling the strings on the spread of this deadly cancer, what other surprises are hiding in our bodies?
It's mind-blowing, right?
Yeah, and how could understanding those connections totally change the way we treat diseases in the future?
That's the million-dollar question.
It is. It really is. So if this has piqued your interest and you're eager to learn more about pancreatic cancer or metastasis or how neuropeptides work in different diseases, we really encourage you to check out the original scientific paper. We'll put a link in the show notes, or you can find it through those online research databases. And definitely explore those reputable cancer research organizations out there. They've got a wealth of information.
Definitely.
And, think about what questions this research brings up for you. Like, what other investigations do you think are important to do next?
Right. What are you curious about?
Exactly, because ultimately understanding these complex mechanisms that are at play in diseases like pancreatic cancer, even though it's really challenging, it can be empowering.
It really can.
And it can fuel that hope for new and better treatments.
Absolutely. And your engagement, your curiosity about this kind of research, it's vital. It's what drives progress.
It really does.
Thank you so much for joining us on this deep dive. We explored some truly fascinating and important findings in cancer research today.
Yeah, it was really cool. As we've seen, that signaling pathway with neuropeptide Y and neuropeptide Y receptor 1, it seems to be this major player in how pancreatic cancer spreads, and it could be this amazing new target for therapies.
It's definitely a game changer. Now, our next deep dive, we're going to be exploring another really interesting topic, and we're using sources that you, our amazing listeners, have shared with us.
Yeah, we love hearing from you, so please keep sending us your sources, your suggestions for future deep dives. We're all ears.
Absolutely. Keep those brains buzzing.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.